Good afternoon, everyone. It is my pleasure to now introduce Professor Dr. Sheeba Vasu. Sheeba is an associate professor at JNCSR, Bangalore in India. She received her PhD from JNCSR in 2002. Subsequently, she was a postdoc fellow at the Department of Neurobiology, UMass Medical School, and Department of Biology, New York University, and also an associate specialist at the Department of Physiology and Biophysics at UC Irvine between 2002 and 2008. She then joined JNCSR as a Ramanujan Fellow in 2009, where she has remained since then. Sheeba's research tackles two important and distinct aspects of circadian clocks, their evolution and underlying neuronal circuitry, using Drosophila melanogaster as a model system. Please welcome her with a huge round of applause.
Thank you very much to the organizers of Horizons 2024 for this invitation, and thank you for all the detailed planning and organizational skills that you have displayed. All of us speakers have been hugely benefited. I especially emailed you guys several times. You've been very patient. Thank you very much. Especially, this is an honor to come and talk about our work that studies circadian rhythms or biological clocks in Germany, because this is really the place where historically the field developed and the original foundations of our field were laid in this country. So I'm especially honored to talk about our work here. I will talk particularly about the work of Pragya Sharma, who's a PhD student who's about to graduate. And I understand, I realize now that my title is somewhat complicated, but I will try to walk you through these terms as we go along. So we are all very, very well aware that geophysical forces very, very strongly influence life forms on Earth, and it's only modern humans that have managed to somewhat insulate ourselves from the very, very predictable changes in nature. And across millennia, life has also evolved systems, biological timekeepers that help us to deal with these very predictable changes in nature. Because of which, we have these entities inside biological systems that time when certain behaviors and physiologies must occur. From this example, you can see that at sunrise, there are some organisms that will seek food and head out of the safety of their homes, while others will do the same at a different, completely antiphasic time of the day. While at the same time of day, there are other organisms that are choosing to come back to the safety of their homes. And this is, even though we might fool ourselves to think that we are not that much affected by these forces, we know now that our biological clocks indeed have major role to play on our physiologies and our behaviors. Okay. We can think about clocks and their influence on our behaviors and physiologies in a very simplistic form. We can think of their existence of these core clock mechanisms that are self-sustained oscillators that can measure the time of day. However, these clocks, being only approximately 24-hour clocks that are in the range of 24 hours, need to synchronize to the external environment, which is a strict 24-hour periodicity. So these core clocks must be sensitive to various inputs, and so different clocks utilize different time information from the outside, including even food, which can reset these near 24-hour clocks and dictate both physiological and behavioral rhythms. In my lab, we are interested in various aspects of this tri-structure. And I must also remind you that these are not really watertight compartments, and we can't really separate these. So these students I've listed out like this simply to talk about the perspectives with which they look at this organization. Okay. But this clock that I depicted in the previous slide here, one must think of more than a clock, but more like the gears and the dials that make up the clock, the cogs that make up the clock. And molecularly, we know over the past few decades, thanks to the work of several people, several labs across the world, that you may recall in 2017, the trio of Jeff Hall, Michael Rosbash, and Michael Young received the Nobel Prize for their discoveries that tell us what this core clock is made up of, which include several canonical core clock genes and proteins whose transcription and translation is regulated via feedback loops, such that the transcription of period and timeless genes are dictated by this heterodimer of clock and cycle proteins, which result in a slow accumulation of period and timeless, which then must heterodimerize and get stabilized by various post-translational modifications, and then enable the entry of this heterodimer into the nucleus and also repress their own transcription, thus completing a negative feedback loop. But this is simply the most basic structure of the clock. Various other loops are part of this core clock, but the beauty of the system is, and the discovery of this, or the construction of this model, is that such entities have now been found starting from cyanobacteria to complex humans. So the basic structure of the existence of transcriptional translational feedback loops forming the core clock is an important discovery, and these are what determine whether an organism will exhibit a nocturnal or diurnal phenotype or anything in between. So now I've brought you to the word waveform, which refers to the timing of either behaviors or physiologies. Going to human beings, do we exhibit variety in waveforms? This is an example of a study conducted again from a German group, Till Ronneberg and Martha Merrow, somewhat dated, but on an European population, asking people via questionnaires about their preferences for the time of day at which they would sleep. And so the mid-sleep timing is plotted, and you can see already that there is a distribution, and very likely single genes do not have large effects, but rather this phenotype is coming from the action of several genes with small effects. So we have individuals, several of us will fall in the middle of this distribution, when we call ourselves the intermediate phenotypes or chronotypes, time types, while there are several of us who might fall in the early or late types. How do these waveforms evolve? And it's not easy to carry out studies concerning the evolution of clocks on humans, and therefore to ask the question of what makes some of us early birds while others are night owls, we turn to this model system, Drosophila melanogaster, which again has been historically one of the models that were used to discover the underlying clock and its molecular machinery. And the fruit fly adult emerges out of its pupal cases through via a circadian rhythm, or it's tightly circadianly regulated, and there's a peak in emergence around dawn. And this rhythm will therefore persist even in the absence of any external time cue. And historically, again, Erwin Buhner and Hans Kalmus from Germany were the first people who studied circadian rhythms in Drosophila. So this rhythm has been very extensively studied and was the basis on which Seymour Benzer and Ronald Konopka first discovered genes which, when mutated, resulted in the disruption of the rhythmicity. In addition to the eclosion rhythm, Drosophila melanogaster exhibits rhythms in several other behaviors as adults. Okay, so using the eclosion rhythm in our group in Bangalore, India in 2002, a set of populations were derived to ask the question about evolution of chronotypes or time types. To do so, we have these control populations which exhibit this very typical morning emergence of adult flies from pupal cases. However, from these populations, when you very deliberately select flies that will emerge during the morning or the transition from night to day, and go on to make them the flies that will contribute to the next generation, and keep doing that over the course of now 24 years and 400 generations, we have a set of flies or populations of flies that exhibit a morning emergence and are referred to now as the early populations. Please remember that we have 4 replicate populations on which we apply the selection pressure. Also, as a contrast, we have flies that are being selected to emerge in the evening, such that now we have a set of populations that we call the late populations that have an evening emergence. And therefore, we have these distinct chronotypes of fruit fly populations. And they are ripe for asking a whole range of questions about chronotype evolution. Coming back to my model, where we have selected for the phasing of an output, the eclosion rhythm, and we are asking, by doing so, how might things have changed in terms of the underlying molecular clock and the inputs and their sensitivity to various inputs. I must also remind you here again that our selection is on flies that are living in a constant 25-degree environment where light dark cycle in a rectangular form is the only time cue that they perceive. Now we can see this is real data, not the cartoon you saw before. So always in green is our control populations with this peak around dawn, and the early populations have now a higher amplitude peak of emergence around dawn, while the late populations largely emerge in the evening. This is under a light dark cycle, and we characterize this waveform of emergence through various metrics, some of which I have depicted here, such as the onset or the peak or the something like a median emergence phase and the offset of emergence. Okay. Interestingly, across several experiments for various reasons, we discovered that if these populations were given temperature as a time cue, things started to look very different. Now, if we performed a similar experiment where there was no light dark cycle, but only temperatures as a time cue, so cyclic warm and cold cycles, we found that these populations diverged even more than the divergence that we saw under a light dark cycle. This was very surprising because we did not expect that anything might have changed about temperature sensitivity to these clocks. But that led us to ask whether something has changed about the organization itself of the clock, which means I must explain to you what I mean by this organization. That takes us back to a model that was proposed in 1959 by Pittendrich and Dahn, which, when they had no knowledge of the molecular clockwork itself, but when people were looking at these biological clocks and comparing them with physical clocks and oscillators, they proposed that the eclosion rhythm is mediated by a pair of coupled oscillators, which consist of an A oscillator, which is light sensitive and is temperature compensated, and is more of and somewhat of a dominant oscillator over the B oscillator, which is light insensitive but temperature sensitive and is more peripherally located and ultimately mediates the phasing of emergence. So we asked whether this system has been changed due to our selection of a particular phase of emergence. And by various experiments by which we altered temperature cycles, kept light fixed, or vice versa and so on, which I will not go into, I want you to take from me for now because this is already published work. We saw that indeed in these late emerging flies, these populations have, it appears, to have evolved a more strong or more temperature sensitive oscillator. So the scales are tipped to the right such that the B oscillators appear to have become more strong. Okay. This work was done by Abhilash Lakshman, who graduated a few years ago. Abhilash also asked, even though we've applied selection pressure on eclosion rhythm, have other rhythms changed? So it's easier to look at activity rest rhythms. It's a more high throughput rhythm. And so Abhilash looked at the activity rest pattern. Surprisingly, the activity rest pattern, of course, its waveform is very different from eclosion. It's bimodal and there are peaks around the lights on and lights off transition. There didn't seem to be much difference across these three sets of populations. Here light and temperature are being supplied. So then we took out light from the mix and asked what happens with temperature. This is not the profile, I'll show you that in a minute. What Abhilash found was that even though overall they may look very similar, again, the late populations have a slightly shifted evening peak of the activity rest rhythm. This is a different rhythm than the eclosion rhythm. Eclosion happens once in the lifetime of the fly, whereas locomotor activity is something that the fly can do almost throughout its adult lifetime. Now, what Abhilash did was to challenge the system a little more, not simply looking at what the activity rest rhythm is like under a warm cold cycle, but giving them a challenge of what some of us have experienced recently, which is jet lag. So either eastward or westward travel of about 6 hours, and I'm showing you only one experiment. So this is what we chronobiologists call activity rest graphs or actograms, and I hope you can see a rhythm in it. So this is a plot across 48 hours, and this is sort of day 1, day 2, day 3 and day 4, where there's a warm cold cycle applied, and hopefully you can see a pattern with which these adult flies are showing locomotion at certain times of the day, stopping locomotion and resting, and doing that every day with a certain phase relationship with the temperature cycle. And now you provide a jet lag, which is delayed warm cold cycle, and you see that flies take a few days before they can now synchronize to this newly imposed jet lagged warm cold cycles. And we asked whether there's any difference when the early and late flies are challenged with this jet lag. And we see that the early flies are not that different, maybe they take a little longer. However, interestingly, the late flies seem to be able to resynchronize or re-entrain to this shifted warm cold cycles at a faster rate. So, along with the increased temperature sensitivity that we saw with eclosion rhythm, we can see that the activity rest rhythm temperature sensitivity has also been altered. Now, with the behavioral and physiological studies that I sort of summarized till now, I hope I've convinced you that selection on phasing for eclosion under a light-dark cycle seem to have altered the clock organization in such a way that both eclosion and locomotor activity rhythms and their clocks have been altered. And it's not only light, but temperature sensitivity has also changed. Now Pragya started to ask the question, what about the underlying molecular correlates of these observed changes? And I will give you this caveat right now, that while I'm very confident of all the behavioral and physiological rhythms and studies that we've examined, because as I told you, all our studies are based on multiple cultures, multiple populations, and we only conclude when we've seen these changes to be statistically significant across populations. We've not been able to do that with our molecular studies for various practical considerations, but I will, at the risk of having to give you a pinch of salt, still present the data because this is a molecular biology conference. So the proposed mechanism for how the clock is sensitive to temperature is a lot less understood compared to light entrainment, which has been typically has been studied for several years, and we know a lot more about the molecular components that control it. So at this moment, what we know about the molecular clock, I will present, and those studies have been usually restricted to looking at the behavior first when there's either a constant cool temperature or a constant warm temperature and a light dark cycle. So in both of these cases, time is in the form of light dark cycles, but the ambient temperature is kept at a constant cool 18 or 29 degrees Celsius, and like us, flies will also shift their activity into somewhat more tolerable times of the day when the temperature is warmer, or into the daytime when the temperature is cool, compared to the preferred temperature of 25 degrees Celsius for the fruit fly. This is accompanied by some changes in the molecular clock, and this is again, like I said, a very recent area of study, and researchers have been looking at what happens with splice variants of the period protein, the period gene, the timeless gene, as well as the clock gene, and this is over the past 5 years, and they have proposed that there are changes due to alternative splicing of these genes, which result in variation in RNA stability, translation, protein integrity, and ultimately possibly functioning of these code clock components. And as I said, this is at this moment still very young, and the field is still exploring how this happens. So with this background, Pragya Sharma decided to explore in particular the timeless gene and variants of the timeless gene for certain reasons. And this is what we know about the timeless gene and its splice variants. There is the canonical TIM long mRNA that is produced, and the TIM short cold and TIM medium forms, as well as the TIM cold forms. And as you might guess from their names, Under 18 degrees Celsius, the Tim short cold and the Tim cold isoforms are highly expressed, whereas at 29 degrees Celsius, it is the Tim medium isoform that is highly expressed. The long form is not that much altered by temperature. This is the overall picture. Additionally, the labs of Michael Rosbach and Sebastian Kadener have and Joanna Chiu at UC Davis, they have all proposed that several splice machinery are altered or probably altered, microRNA machinery probably altered under these conditions. Okay. So with this background, what Pragya did was to examine these splice variants in our populations. But I should remind you again that the previous studies have mostly been looking under conditions of constant 18 or constant 29 with light dark cycles as a time cue. But we thought it is really important for us to ask what happens under cycling temperature, because light, timeless, is in fact a very superbly light sensitive protein, and timeless can in turn affect its own transcription, and therefore it was important for us to keep light out of the picture and ask under purely temperature cycles what's going on with the mRNA oscillations. So we wanted to look at that. We also wanted to bring in light, dark and temperature cycles in sync, which is probably a bit more realistic. It's not like it's constantly cold in the winter and constantly cold in summer, and that there is also an oscillation in temperature. Addition to that, we also realized that in fact it's not that light and temperature cycles are perfectly in sync, and you've all noticed that just before dawn is in fact the coldest time of the day. And so we did a somewhat more realistic situation where light and temperature are out of sync, such that the warm temperature begins to rise 6 hours after lights have come on. So under all of these conditions, we've examined the splice variants for timeless. And so we've looked at these in the control populations as well as in our chronotype populations, and we also examined them under these three regimes. And the logic for looking at these two regimes was also because we had some behavioral data that suggested that if you compare between the in-sync conditions, so I know this is a lot, so this behavioral data is for experiments conducted comparing in sync versus out of sync light, dark and temperature cycles. And under this particular combination of light, dark and temperature cycles, we had seen that there is a distinct difference between the late and the early populations in how their waveform plays out. And that's part of the logic for looking at the splice variants under this particular conditions. Okay. So with these questions in mind, we did these experiments where we are sampling for these mRNAs under each of these three conditions, and we are looking for each of these three or these four variants. So I'll quickly tell you about what the expression profile looked like for control, because this is again unreported by anybody before. So it was important for us to see how the TIM mRNA might oscillate if there was only temperature as a time cue, and this is what the waveform looks like. And since we know that under temperature cycles there is a shift in the phase for the late populations, we were curious to see how our early and late populations behaved, and we do see a bit of a shift here again in the late populations in when the timeless levels start to fall. So it's kind of matching what we can see in behavior. There's also a difference in the amplitude of the oscillation, and there is a nice similarity in the direction of the shift for activity as well as the mRNA oscillations. Okay. So then we asked, what about these variants in the splices, in the spliced forms? We look at the cold induced variants under temperature cycles, and our expectation was that we would see differences in the cold part of the day, which we do see a slight shift in the late populations. But we also see a difference in the early populations at a time of day when the cold variant is not supposed to be expressed. We don't have a good explanation for why that is happening. We see again a difference or a change in the phase in the direction of our activity behaviors. We also see a change in the overall levels of the TIM cold isoform in the late populations. Then we asked what happened to the warmth-induced variant under temperature cycles, and here we see that the warmth-induced variant in the early populations is significantly higher than the controls and late. So overall we can see that the changes in the behavior that we had observed is correlated with differences not only in the overall long form of the TIM mRNA, but also in the variants of TIM, both the cold and the warmth, although we don't have good explanations of how that converts into changes in behavior. We also looked at, as I said, these in phase and out of phase light, dark and temperature cycles. So what I am plotting here, the solid curve, is when the light and temperature are out of phase with the controls as compared to the in phase situation, and as expected there is a right shifted profile of overall timeless or the long form, and there is a difference in the phase for these two. That does not translate in the early populations because the peak, if at all that is the peak, is the same both under the in sync and out of sync regimes. On the other hand, in the late populations, we can see that the in sync and out of sync regimes have very different profiles in the mRNA oscillations. This also correlates nicely with what we have seen in the activity phase, where the late populations have a distinctly delayed phase when you have an out of sync light and temperature cycles. So there is a drastic change in the phase of peak expression of timeless in the late that is unaltered in the early, and there is an overall divergence in the early and late mRNA expression pattern, which is matching with what we have seen with the activity pattern of the controls. Then we also looked at the variants, the TIM cold under in sync and out of sync. Here, as expected, there is a shift on to the right, but we could not make any major conclusions. They look very similar. The only things that I can draw your attention to that looks different is that at this point or at this transition point where only the light dark transition occurs while temperature is kept constant, this TIM cold variant is particularly sensitive in the lates and in the controls, and appears to be insensitive in the early populations. We also looked at the TIM medium variant, and in the TIM medium variant again we can see a shift in the phase both for the early as well as for the control populations, and that shift is gone in the late populations. So overall we can see variations not only in the behavior but also in the underlying mRNA variants in these three sets of populations. So I hope I've been able to show you some evidence for the evolution of altered clock machinery at the level of splice variants in responding to temperature cycles, which is new, and we are curious to ask whether the splicing machinery itself has changed in these populations, and we were beginning to test these in our populations. Moreover, I must point out that the studies that we have done are in the adult fly, whereas the phasing of adults emerging out of the pupal cases is where our selection pressure is being applied. So it would really be important for us to look at those stages in the fly to ask what has changed there. We have been very simple about how we've designed our regimes. However, most of the studies that have been looking at this are in the context of seasonal variations. Changes in thermoperiod and photoperiod would be the most relevant conditions in which we must be testing these or asking questions. So with this, I would like to thank the people who have contributed to various projects in our lab, including funding from my own host institution as well as several national agencies, and our national and international fly community who have been very generous with sharing several reagents. This is Pragya here in the middle, who has recently completed her colloquium, which is sort of preparing for writing up her thesis, and the various members of the lab, including summer interns, are in this picture. This study was initiated by Professor Vijay Kumar Sharma, who is no more, and so it was very prescient of him to set up these populations with the help of his graduate students Shailesh, Kaustav, Nikhil, Abhilash and Arijit, all of whom have contributed variously over the course of these years. Most recently Pragya, and more recent students Manasi and Neeti will hopefully carry out more investigations on these populations. So with this, I thank you for your attention, and I will be happy to take any questions you may have.